Hello,
Hello.
my name is Parsa. I'm in my third year of health sciences at McMaster, and I'm joined by
Olivia. I'm also in my third year of health sciences at McMaster. Today we're going to talk a little bit about anatomy. This is one of our final deliverables for Health Sci 3F03, which is Inquiry into Anatomy and Physiology. Yes? It's also
Yes.
Physiology, right?
Yes.
Okay.
Inquiry Anatomy, if you didn't know, is a new course developed at McMaster. It seeks to not didact information in anatomy, but it adopts that inquiry style where students in the course are able to research topics that are of interest to them, and throughout the term you can research different topics. But one key difference is that throughout the term you're attempting to create different connections between your topic and what you research and other people's topics.
Yeah, and I think this is a topic like anatomy that really lends itself to something like inquiry because it's just so vast and also so niche at the same time. There's little pockets of research everywhere, so it kind of lets you go as broad or as deep as you want, and everything connects to everything else. And so I think the whole idea of inquiry, where you're sort of exploring this like web of knowledge, this is a good topic for that.
Yep, I think especially for connections. There are some connections that definitely make a lot more natural sense. Like I think like diabetes, kidneys, things like that. Me and you went for a more untraditional connection,
Yeah.
I would say, to filtration organs. So just some intro. I was really interested in the liver and fatty liver disease. And
I was really interested in the placenta, and especially like the effects of the placenta and like placental dysfunction for the parent versus like placental effects on fetal development.
mm-hmm. Yeah, I think it also makes sense just before we go into our research, maybe talk a little bit about why we took the course, why we chose our topics. I remember I took 2 FO3 and 2 FF3, our second year anatomies here at McMaster, last year, and I felt like it covered a lot of topics, but there were still some things which weren't taught in as much detail, which I was still interested in. There was a course I couldn't take this term, so I needed a replacement course, and I remember I couldn't take inquiry anatomy because I had an anti-rec for it. I asked Dr. Bridgewater, and he was able to let me in as long as I studied something which I hadn't done before, which is precisely what I did. So I was interested in learning a bit more about the liver when its metabolic pathways goes wrong with fatty liver disease and repro as well. So I feel like this was a really good connection to make.
Yeah. No, I kind of came into the course with no expectations, and then when they were like, what are the things that you're interested in, I sort of bounced around a lot at first. I was like, I'm interested in blood, I'm interested in blood pressure, repro is cool. I feel like at the end of second year anat, everything gets so busy that even if they're dedicating, like they dedicate quite a few lectures to repro, but like I wasn't really.
Like 5 lectures, they're all crammed.
Yeah, it's a big topic, and like I wasn't super into learning it in depth. I was just trying to get by and finish the course by that point.
Mm-hmm.
And I think a lot of the focus of that repro was on fetal development, how does baby get made, get born, and less on like what that experience is like for the parent who's like growing a whole person inside of them.
Yeah.
And so I kind of wanted to take that lens and see how pregnancy experiences can kind of vary like that, what goes wrong.
Mm-hmm. I guess your topic, placenta preeclampsia, good way to look at that. I think it's also interesting to note the liver and placenta both being not only organs related to filtration, also have a fetal origin, right?
Mm-hmm.
Just getting to some of the connections. When the fetus's liver is still not fully formed, it doesn't make sense to shunt all the blood through the liver.
Right.
So the liver and placenta have their first connection really when the umbilical vein coming off the placenta bypasses the fetal liver, becoming the ligamentum venosum, or I guess, sorry.
At the time, it's the ductus venosus.
Yeah, ductus venosus, draining into the inferior vena cava. And then that then becomes the ligamentum venosum.
Yeah.
But really interesting how those organs are sort of connected physically.
Or almost not connected, right? Because
Yeah.
it's going straight.
Yeah,
by.
yeah, not connected. It's interesting. These vessels people once thought could be reopened because of portal hypertension, which we'll talk about. So when those vessels in the liver sometimes get occluded in cases like cirrhosis or end-stage liver disease, people thought the blood would pool and recanalize things like the umbilical vein, which would then be the round ligament at the time. But that actually is a myth. So no angiography study has ever proved that.
Wow.
What really happens is the blood tends to pool in like the periumbilical veins and other superficial like epigastric veins.
I wonder where they came up with that then. Like, someone was just like, I have an idea.
Yeah.
And then everyone was like, cool. We agree.
maybe it was poor, like maybe they did like a CT, and they were, like, and they said, this looks like that's the course it would run. Maybe they run close to each other. Yeah. If you don't mind, would you like to tell me a bit more about like some of your original research on, what is the placenta? How does it normally
Just generally,
form?
The placenta's really cool because it does form. It's not something that we're born with.
Mm-hmm.
But basically when you look at the regular anatomy of the uterus, you have all these different layers of the uterus. Like the outermost layer is the myometrium, and it's like the muscle, but then inside you have the endometrium, and there's 2 layers to the endometrium. There's like a basal layer, and then on top of that, so deeper, close to the lumen, there's a functional layer. And basically at the beginning of pregnancy, you have your blastocyst, and the blastocyst has those 2 parts. One part, the trophectoderm, it becomes the trophoblast, and it has these cells that bury into that functional portion of the endometrium and remodels all of the arteries and the extracellular matrix. And so that functional part, this is what I was arguing with the surgeon about.
Is it just the endometrium?
No, it goes further, but he was saying the endometrium isn't the placenta, but the functional part of the endometrium becomes the decidua, and that's the maternal portion of the placenta. So at that point, it's not considered the endometrium anymore, but that's what it used to be. So the functional endometrium, which is the lining that gets shed every month, in menstruation, that part becomes the decidua when it's remodeled by the trophoblasts.
Right. So there's these 2 parts. One's the decidua from the mother, and the other part is the fetal contribution.
Yeah.
Which is called
the chorionic plate.
Mm-hmm.
So yeah, it's
chorion.
2 sides.
Mm-hmm.
You have the chorionic plate and the basal plate.
Mm-hmm.
So the basal plate is attached to the maternal endometrium, the basal part of the endometrium, and then the chorionic plate is attached to that. So it's a sandwich.
Right.
Yeah, and it's that chorionic plate that has the umbilical cord attached to it, and when you look at it, that's the part that has all of the vessels running along it, which is pretty cool.
Mm-hmm. Mm-hmm. What about, how does it infiltrate those, and then the vessels sort of infiltrate, right?
the trophoblasts infiltrate.
Infiltrate the spiral arteries?
Yeah, so the spiral arteries they're already existent. Like, they exist in the endometrium and, like, a little bit into the myometrium. And the trophoblasts kind of, like, eat into the, like, spiral arteries. They strip away all of this material to widen up the spiral arteries. And, like, doing that allows more blood to flow into the placenta. It, like, creates low resistance.
Right. What would you say are the functions of the placenta?
I think people normally talk about it as, like, the interface for nutrients exchange. But the placenta also, it creates all of these, like, hormones and, like, cytokines. I think it creates almost every single cytokine known, which is, like, wild. I don't remember where I read that. It's somewhere.
Released into fetal circulation? Maternal circulation?
Maternal circulation.
Oh.
So, I want to say this. Okay, so you have, like, the chorionic plate, and then you have the basal plate, and coming off of the chorionic plate are these villi, like villus trees, basically. And each villus tree contains all these fetal vessels, but the outer layer of that villus tree is syncytiotrophoblaст.
Right.
And the syncytiotrophoblasts are, like, really responsible for, like, creating hormones, creating cytokines, and, like, releasing them out into the intervillus space. And so all of that's the mother's bloodstream. So it releases it mostly into the maternal circulation.
Right. Interesting. I guess especially in inflammatory conditions, you get more of that cytokine repertoire.
Yeah.
Like, I know preeclampsia is a highly inflammatory condition. Right?
Yes.
The placenta screaming.
Yeah.
Is how I would describe it.
And we'll get into that later. Do you want to talk about the liver?
The liver?
Liver normally?
Yeah, I can talk about... I'm assuming our audience has a basic understanding of anatomy and physiology, but just for refresher, liver is this, like, major metabolic organ, right? Taking blood from the portal vein, from the intestines, which is very nutrient-rich, and essentially filtering it. Not only nutrient metabolism, but also drug metabolism, and waste excretion. At the same time, it's receiving oxygenated blood, because remember, the blood from the portal vein is very deoxygenated, having just gone through the intestines. So it's also getting 25% of its blood, which is oxygenated from the hepatic artery, all going into this little entrance at the back of the liver called the porta hepatis. Almost all the functions of the liver are done in these cells, which comprise the majority of it, called hepatocytes. Remember, anything with hep at the beginning is related to the liver. So these are just fancy words for liver cells. In terms of nutrient metabolism, some things you might already know, it converts glucose into glycogen, right, the storage form. It's also the only place, aside from the kidneys, that undergoes gluconeogenesis.
Oh, I didn't know that.
Yeah, the kidneys also do it, but it converts things like proteins and amino acids, even pyruvate, these different metabolic substrates back into glucose when your body doesn't have it, even when it doesn't have glycogen, which is super cool. Also the only place which creates ketone bodies from fatty acids, and you need that in the starvation state when you're in ketosis. That's when your liver is just churning out these ketone bodies to be used by places like the brain especially, which can run off ketone bodies. And it also packages and produces new triglycerides, essentially, from fatty acids. Creates these triglycerides and sends them out in lipoproteins, like VLDL or LDL. You might have heard of these, like low-density lipoprotein. These are essentially transporters for getting that triglyceride to other places in your body, especially adipose, right? Adipose tissue. So a lot of nutrient metabolism. Also produces bile, right? It's a really common mechanism for it. Bilirubin is from heme breakdown, from your hemoglobin, is also churned out in bile. This happens in the liver. I think a lot of this was covered in anatomy, but just more detail on some of these processes. The liver detoxifies drugs, so adds hydrophilic groups to hydrophobic molecules so that they can be essentially excreted out through the kidneys. Also produces a lot of important proteins. so coagulation factors for clotting, albumin for retaining fluids, important for oncotic pressure, just keeping the fluid in your vessels. I think the coolest thing I read about the liver, which was completely new to me, might be new to you, is the sort of the zonation of your hepatocytes as well. So you might not notice about the liver, but its functions is really separated out in terms of where that hepatocyte falls. So between the central vein, which is taking blood back to the heart, and the portal vein, which is bringing blood to the liver, there's this capillary, and along it is a line of hepatocytes and some endothelial cells in between the blood. But the hepatocytes, depending on how far along they are, perform different functions. So the periportal region, being close to the portal vein, does a lot of oxidative metabolism, beta oxidation, gluconeogenesis, as well as producing bile. Whereas as you get further down to the central region, it does more detoxification and biotransformation of drugs, as well as ketogenesis, glycolysis, and lipogenesis. So you see the different functions as you get from one side to the other. Very cool, like, organization of the cells as well.
I wonder how that, like, developed.
I'm not sure. That's a good question.
A further area of research.
Might have to do with the signaling in different areas too. Do you see any sort of organizational difference with the placenta?
The placenta is, when they talk about, like, the anatomy of the placenta, it's pretty much like disc, like, and then in between is the intervillous space.
Mm-hmm.
But actually, when you get into it, you have the villi that extend off the chorionic plate, right? And all of those villi are covered in the syncytiotrophoblast.
Mm-hmm.
And the syncytiotrophoblast, it uses up, like, I think 40% of the oxygen of the fetal placental unit, which is crazy because it's actually creating, like, a lot of things, like steroids, peptide hormones, cytokines. But actually, because it's using up all of that oxygen, normally this would mean that the fetus would be getting less oxygen.
Mm-hmm.
So it's organized that the areas closest to the vessels, to where, like, oxygen exchange would occur with the maternal blood circulation, are actually very, very thin. Like, the syncytiotrophoblast cells are very thin, and they don't have organelles. And so those cells aren't creating, like, steroids or, like, hormones to release into the bloodstream. But the ones that are further away, where the fetal maternal blood or, like, oxygen and nutrients exchange would happen, the ones that aren't close to that are the ones that are, like, using up more oxygen to create stuff. So it's organized to try to not impede as much on the oxygen exchange, to, like, let the fetus get as much oxygen as possible.
Yeah, it's like trying to spare the oxygen diffusion in the areas that's the closest to where the fetus would be receiving it. It's very interesting. I guess it's similar to the liver. It's like zone 1, the closest one, that's doing most of the nutrient metabolism, trying to do the things like especially like oxidative metabolism. So tell me a bit more about when things go wrong. What happens in the placenta when things go wrong?
Yeah. So one of the biggest things that I was looking into with the placenta specifically was preeclampsia. And I think, like, over 100 years ago, preeclampsia was described as, like, a disease of theories, because even though they know, like, there's these things that are probably related to preeclampsia, they know, like, okay, preeclampsia, it has a brain component because there's seizures and coma once you get into eclampsia, and it kind of has, like, a blood component because of hypertension. And it has a kidney component because of protein in the urine. So initially they didn't really know where preeclampsia was coming from, and that's what drew me to it, because it's still, I think, a very mysterious illness. And even though there's so much research on it, they still don't know exactly what causes it. So basically, preeclampsia is determined by two main factors, and that's sudden onset hypertension around 20 weeks of gestation, and then protein in the urine. Yeah, and it happens in about 3 to 5% of pregnancies, so that's not like uncommon, I would say.
And just for some background, preeclampsia, remember that's when it begins with aberrant placentation, right?
Yeah. So yeah, basically it's probably caused by improper trophoblast remodeling of the spiral arteries. And so basically your placenta isn't getting enough blood.
Wow.
But they think that there's probably something in between, because you have placenta, or like improper modeling, and then you have preeclampsia. And there has to be something in between, because other times there's issues with remodeling of spiral arteries and like improper placentation, and it doesn't lead to preeclampsia. It leads to like other issues, like fetal growth restriction, but without maternal complications. So there has to be some sort of other factor that distinguishes it into preeclampsia, and that's what they're still looking for.
Wait, what's the second hit?
Yeah.
Almost. Would it not be ischemia?
They think maybe it's the body's response to the ischemia.
Okay.
So if your placenta isn't getting enough blood, and then it kind of flips out and like releases all of those like inflammatory factors and cytokines, and that damage causes preeclampsia maybe, like in connection with the spiral artery remodeling issues. Because like you get spiral artery remodeling problems, and if you don't have that like widespread inflammation, it's not preeclampsia. They do Doppler studies on people, and they look at like, okay, you don't have properly remodeled spiral arteries, and not all of those cases end up becoming preeclampsia.
Right.
So it's not specific to preeclampsia. That's why there has to be some other factor, but they don't know what it is.
So you can have poor remodeling of the spiral arteries, which means that you may not be getting sufficient blood flow to the placenta. That's maybe, probably not guaranteed. But without the inflammation, without the immune response, it's not really preeclampsia, because you don't get the systemic effects.
Right. Without the systemic inflammation,
Right.
It's not preeclampsia.
That's really interesting. I wonder.
So yeah, what causes that other?
Yeah. It's a little black box.
Yeah. They're still trying to find it. They don't know what it is.
Could be different subtypes. I think that's like it's probably a big thing now with a lot of diseases, different subtypes. Like one subtype is an ischemic response. Could be other subtypes.
Yeah. Insulin issues.
Yes, for sure. Also the same with fatty liver. A lot of metabolic syndrome causing a lot of issues around the body.
I didn't even know metabolic syndrome did so much. Like, I mean, we'll get into it later.
Yeah.
There's a lot going on with that.
Yeah.
Do you want to introduce me to fatty liver
Fatty liver,
disease?
for sure. Okay. Fatty liver disease, there's different types. Non-alcoholic, alcoholic fatty liver, even viral-induced, like hepatitis. With fatty liver disease, it affects about 25% of people in the U.S. It begins with just simple fat accumulation in the liver. It's called steatosis. So basically your hepatocytes aren't supposed to carry a lot of fat in them. The liver's not a fat storing organ. But what happens is, because of so much fatty acids in circulation, usually about 60% of the fats come from just circulating free fatty acids. They sort of accumulate in your hepatocytes and create these lipid droplets.
Okay.
At first it's benign though. It doesn't cause issues or inflammation or anything. And you might get a little bit from your diet as well, 15% from the diet, another 25% from de novo lipogenesis. So that's just liver, that's just hepatocyte producing fats in the endoplasmic reticulum. And there's a bit of beta oxidation, which is the breakdown process of fatty acids, also happening in the mitochondria, which can try to account for some. But it's also breaking down a little bit of those fatty acids.
Mm-hmm.
Now, over time, the problem with fatty liver disease is, because this concept of lipotoxicity, the lipid buildup actually ends up causing damage to your hepatocytes and the liver. So you end up going to this condition called NASH, or Steatohepatitis, really.
Right.
And that's when your liver almost like swells up a bit, and these fat pockets might get a bit bigger because it's all inflamed. So the hepatocytes having so many lipids, it's trying to break it down now using beta oxidation. But the problem is almost every step in beta oxidation produces reactive oxygen species as a product. So you're creating these reactive oxygen species, and those are just these oxygens that are free radicals. They can react with different things like DNA, like the cell membrane, even the mitochondrial membrane. And this causes, this can activate cell death pathways, cleaved caspase 9s, just apoptosis, even necrosis. So that's not great. At the same time, triglyceride production in the ER leads to ER stress if there's too much of it happening at once. And ER stress, just buildup of those metabolites, also signals for apoptosis. So you're getting a lot of these apoptotic signaling pathways.
Mm-hmm.
At the same time, Kupffer cells, these are like resident macrophages in the liver, are reacting with the excess fatty acids as well. So they're causing inflammation, releasing TNF-alpha. So you can see inflammation plays a really common role in both of our diseases.
Yeah.
Just that response.
Yeah, I mean, one of the big things about preeclampsia is you have the ischemia and the hypoxia causes the release of all of those cytokines from the placenta, and they like travel because it's those syncytiotrophoblasts that are like creating those. So it gets into the maternal blood supply, and it just travels through the whole maternal system, and like all of those like IL-6 and like TNF-alpha, they cause like endothelial damage, and then that's like swelling. It's like micro angiopathies, where you have like damage to the capillary beds, and what's the other one where like clotting issues, hypertension, because like your endothelium, it can't dilate properly. Yeah, it's wild how much inflammation does, and like how terrible it is for you on like a full body level. Like it affects everything.
Yeah, I think big difference is in NASH, it's very local inflammation. With preeclampsia, you're really dumping these cytokines into the bloodstream.
Yeah.
It ends up affecting organs like the liver.
Yeah.
You know, there's HELLP syndrome, right? Very common occurrence with preeclampsia, and I've read a bit about it in the context of the liver. You might know more about it in a systemic context, but that's also a big issue. We can touch on that in a bit.
Yeah.
I think the problem with fatty liver, though, I think it becomes an issue when you get into cirrhosis, which is when it's not just inflammation anymore, but the liver sort of shrinks down a bit because it now has so much fibrosis. So over time, with all these inflammatory cytokines in the area, and so much cell death, you need to replace that parenchyma with some sort of tissue, and it ends up oftentimes being either like giant lipid droplets in enlarged hepatocytes, or activated stellate cells, hepatic stellate cells, these almost like fibroblasts that are just secreting a ton of extracellular matrix. So you end up getting a lot of fibrosis, and the problem with fibrosis is it's essentially, for one, it might be blocking the path between the hepatocyte and the capillary, because hepatic stellate cells sort of lie in this dead space, called DESI space, between the hepatocyte and the capillary. So releasing the proteins there. and also it impedes the ability for blood to flow and for your vessels to dilate properly. So it's increasing essentially the tone of the vessels in the liver.
Right.
So then you get this thing called portal hypertension, huge issue with liver, and probably the biggest cause of death, because I think we touched on it earlier. All your blood sort of gets pushed back. You're also putting like a clamp on the system, and it fills up those paraumbilical veins and vessels, and you get this condition called caput medusae. So you're getting these like almost like varicose veins running across your stomach, also on the insides, which can cause internal bleeding, like on the esophagus. Yeah. You're also reducing your ability you have less liver cells to produce things like albumin, so you have lower oncotic pressure holding fluid in, so you get edema, especially in lower extremities. I think ascites is really interesting. It's a symptom where you get like fluid accumulation, you get this like giant beer belly, sort of like the edema.
Wait, do you have pictures of the like the effects of portal hypertension?
I have pictures. Let me pull it up. I have pictures of caput medusae. We can pull this up on the podcast in a second.
I'm kind of excited because I've seen you talk about it, but I've never. Oh my god!
I thought I showed you.
No, I've only ever seen you like draw it on the chalkboard.
No.
That's
It's really horrible for
wild.
these patients because these like
That's
giant
gotta hurt.
vessels. And on the esophagus too, like esophageal varices. Yeah. No, super interesting. Also very, very sad. And you know what? Beta oxidation actually picks up in NAFLD, like non-alcoholic fatty liver disease, but it doesn't account for the amount of circulating lipids. Now, I think this is where we found a really interesting connection. We talked about metabolic syndrome.
Yeah.
Fortunately, with non-alcoholic fatty liver disease, it seems like one of the main causes is metabolic syndrome. I know metabolic syndrome plays a role in preeclampsia. How big of a role would you say it plays?
I would say a pretty big role in terms of risk for preeclampsia. So they talk about like if you have like obesity or insulin resistance, diabetes, all of those are really big risk factors for preeclampsia, but not necessarily the cause of preeclampsia. So having like a condition like metabolic disease will increase your risk, but you can have metabolic disease and like not develop preeclampsia. That's what makes it so hard to find what's that second factor beyond improper trophoblast remodeling, you know? Because they have all of these risk factors, but they don't have any factor that in every case always causes preeclampsia.
It could just be not one cause, right? I think insulin resistance could be one. Sometimes it's insulin resistance coupled with ischemia, right?
Right.
Sometimes coupled with like gut microbiome issues,
Right.
even,
Yeah.
right?
So, yeah.
Even fatty liver disease, you're getting more like because fatty liver disease affects how your intestines permeability, so you end up getting more almost like bacteria in your bloodstream, especially like endotoxin, LPS from bacteria. That can circulate, right, and cause all sort. It's causing an inflammatory response, right? Even in the placenta. So.
Yeah. So maybe it's that inflammatory response that's like important as that like second factor. Yeah. Because what do all of these like conditions have in common? In the end, comes down to inflammation, right?
Yeah. So it's almost like
metabolic syndrome it's associated with just low-grade systemic inflammation.
I'll say the one thing that has to be done is poor remodeling, right? Yeah. What did you find with insulin resistance and metabolic syndrome?
I was going to ask you that.
Oh.
Do you want to go first?
I can go first. So I'm sure you know insulin is the hormone produced by your pancreas to stimulate your cells to take up glucose when your blood is hyperglycemic. Also tells your adipose tissue to make more fat and stop breaking down the fat, and tells you glucose. So it tells your liver to convert glucose to glycogen as a storage form. And it can be caused by a lot of different things, right? Like genetics is a big factor, but also poor diet and obesity a lot of the times interferes with signaling.
Right.
Almost like receptor desensitization. You just see less insulin receptors as one of the major causes of insulin resistance, which could be due to hyperinsulinemia. There's too much insulin in the blood. In the liver, the main reason why insulin it's a very direct correlation, and it's the fact that with insulin resistance, your adipocytes are less likely to stop breaking down fat. So even though there's a lot of insulin in your bloodstream,
right,
you'll continuously break down fat and not able to store it properly as triglycerides.
So interesting.
And yeah, you just have this like large outspur of free fatty acids in your bloodstream, which go and deposit in the liver. Eventually, as your body releases more and more insulin, you might end up depositing it as triglycerides, but it's just a delayed response. So every time you're delivering this load of fatty acids to the liver, and the liver doesn't know how to handle it well, and hepatocytes are just predisposed to taking up that fatty acids and storing it as triglycerides. At the same time, you have reduced ability to create VLDL, so those transporters, those lipoproteins that take the fatty acids, convert them to triglycerides and send them out. So they're just being stored as lipid droplets instead in hepatocytes. And without knowing what to do with them, you're trying to break them up beta oxidation. You end up getting reactive oxygen species. You try to convert them into triglycerides to send them out as well, and that's leading to ER stress, and you have less VLDL. So everything is going against you. Also, insulin resistance and also leads to reduced beta oxidation in hepatocytes, so reducing the rate at which the fatty acids are broken down. And given that insulin resistance sometimes correlates with obesity and just diet,
right,
I think it's safe to assume that increased fatty acids from your diet also has a tendency, like I said, that 15% comes from your diet and can accumulate in the liver. So not just correlations between insulin resistance and fatty acids and fatty liver disease.
Yeah.
Even endothelial cells, but I think you have more on that. But I know it disturbs the ability of endothelial cells to produce nitric oxide. Right? But anyways, you can tell me a bit more about preeclampsia and insulin resistance.
Yeah. So there is like that big connection between metabolic syndrome, like insulin resistance, preeclampsia, and it's really mostly in the context of that endothelial dysfunction. So basically, insulin, it's angiogenic, so it really promotes like the healthy remodeling of the spiral arteries and the development of all of those new blood vessels within the placenta. So if your body isn't like recognizing the insulin because it has less insulin receptors, like they've been internalized or downregulated or whatever, then you're not getting that effect from the insulin, right? The angiogenic effect. So normally your insulin stimulates the insulin receptors and like activates them, and it starts this AKT signaling pathway, and that pathway eventually trickles down to increasing your eNOS expression and activity. So you're getting more NO, and it's kind of hard to talk about this without talking about nitric oxide first. But basically, nitric oxide plays such a big role in vasodilation, especially in pregnancy and in the placenta. So if you're not responding to insulin, you're not activating that pathway properly, and so you're getting less eNOS expression and like NO production, and so you don't get the full vasodilative effects that you need for proper function of the spiral arteries, so less blood flow.
Is that related to feeding the placenta or hypertension? Just to make it clear.
Both.
Oh, okay.
Yeah.
You just get overall increased tone of your blood vessels throughout, right?
Yeah. And like basically there's 2 types of insulin receptors in the placental vasculature. So usually the first insulin receptor, IRA, has a higher affinity for insulin, but basically when you're activating IRB, it causes like an inhibition of the AKT pathway. So normally you're activating AKT pathway and it's causing like eNOS production, but for whatever reason in like insulin resistance, you get less activation of insulin receptors, and the ones that are being activated are IRB instead of IRA. And IRB runs that inhibitory loop for AKT expression, like the pathway and eNOS production.
Right. AKT increases the expression of eNOS.
Yes.
Stimulated by IRB receptors.
IRA.
Sorry, IRA receptors.
Yeah.
IRB downregulates AKT.
Mm-hmm.
Right. That's interesting. And
It's
eNOS is creating the right amount of NO.
Yeah. It's the endothelial one.
Right. And you also have iNOS, right? That's less good. Do you see iNOS upregulated? Because for the liver, it's very similar. We have eNOS-derived nitric oxide from LSX, or liver sinusoidal epithelial cells. These are sort of protective against disease. But then when you get that shift to iNOS-derived NO, you end up getting these large amounts of NO instead. Like iNOS produces like a huge burst of NO,
right,
which ends up not being used and actually creates this like reactive nitrogen species like peroxynitrite that causes its own host of inflammation. And iNOS is produced from inflammatory cytokines in our case, or bacterial endotoxin, because like I said, you have this like disruption in the intestinal epithelia. So I was wondering if you also see that same shift between eNOS to iNOS leading to...
I think most of the research about eNOS, iNOS, it's more focused on a lack of eNOS production in preeclampsia. There's a little bit about iNOS, but they normally talk about like just not having enough eNOS. They use iNOS in other parts in pregnancy, like especially for cervical like ripening right before labor, but I think it plays less of a role in like systemic inflammation.
I see. But you still see that like drop in eNOS. I think that in itself causes a lot of issues. It seems like, I know when there's too much of it isn't great. It's creating more inflammation through reactive nitrogen species, but in small amounts it actually helps. Maybe I think I even read that it inhibits the inflammatory activation of Kupffer cells.
Oh, really?
Yeah. So in small amounts it's helpful. Too much of it probably creates a balance towards reactive nitrogen species reacting with your DNA membrane, causing damage in that way. Very finite balance.
It's a very, very delicate balance. And I think especially in pregnancy and in the placenta, because like the placenta doesn't really have any like autonomic innervation. Like when the trophoblasts are remodeling, they're stripping away a lot of nerve tissue, and so like as a result you're not really getting like innervation signals that tell your vessels to like dilate or to contract. And so it really depends on like the more autocrine paracrine signaling, which NO does, right? So it plays like an even bigger role in the placenta in terms of like keeping the vessels dilated and also keeping the myometrium of the uterus relaxed and like stable, so you don't get like contractions too early.
Right. That's just a lot of like local control. Has its own brain.
Yeah.
Placenta's a brain. That's really interesting. So we jumped into nitric oxide for a bit. Let's go back to insulin resistance. You were talking about how insulin resistance is angiogenic, right?
Yeah.
And insulin is angiogenic and helps promote the formation of those blood vessels, which is important during placentation. I think we left off there.
Insulin has like a few different functions like that. Okay. So it's angiogenic, but also its main role of like uptaking glucose is also important, because when you have hyperglycemia in the blood, that reduces trophoblast activity. And I read this really interesting article that showed like a dose response, where like basically the more hyperglycemic the blood was, the worse the trophoblasts like performed. And I was like really cheering for the trophoblasts. Like, I don't know, I felt so like deeply connected to them when I was reading the article. I was like, no, they're not working as hard as they should. But yeah, so basically, the more glucose you have in the blood, the more the trophoblast activity is inhibited to like remodel the spiral arteries. So you have angiogenic insulin function, and then preventing that inhibition through hyperglycemia. But then also insulin runs a lot of signaling pathways. And like basically, normally insulin signaling will preferentially activate the protein kinase B AKT signaling pathway, and that eventually activates eNOS production, which is so important because the placenta doesn't really have like a lot of nerve tissue to regulate blood flow, so it all comes down to NO. And normally your insulin, you are activating insulin receptor A, and it causes the AKT signaling pathway to like click into place, and you have increased eNOS expression, which causes like NO creation, and those effects cause vasodilation. But basically, when you have insulin resistance, you're going to have insulin receptor B preferential activation. And so insulin receptor A preferential activation causes eNOS creation and AKT pathway activation, and insulin receptor B preferential activation inhibits those systems. So you're not getting as much eNOS or NO, and then you don't get the vasodilative effects. And that vasodilation is important in terms of making sure that your placenta is getting enough blood in the first place. And so if you combine that with already poorly remodeled arteries, they think that that could be kind of a contributing factor to preeclampsia.
That's really interesting.
Mm-hmm. Yeah, I didn't realize what a big role insulin actually plays in the body, besides just like glucose uptake.
Yes, very interesting. Are you okay shifting over a little bit to maybe like diet, how that affects everything?
I'm very excited to shift over to diet.
Okay, because I think the liver is a really interesting place, given how it's, this first pass metabolism thing passed through the liver first. I think most interestingly, when I looked into some sugars, it seems like high-fructose diets are a big contributor to things like fatty liver disease. Fructose is an interesting molecule. I'm sure if it came up at all in your research. But unlike natural sources like fruit and stuff where there's also fiber in there, when you drink something which is sweetened with something like high-fructose corn syrup, very, very dense amount of fructose, and fructose is metabolized a lot faster than glucose. It's phosphorylated almost instantly. Fructolysis is practically unlimited. It's not regulated by any like a main rate-limiting enzyme, unlike glucose.
Mm-hmm.
So the thing is, your body depletes the phosphate so fast to try and get the fructose metabolized that, because remember, you have to phosphorylate it first when it comes into the cell.
Right.
You create glucose 6-phosphate, you create fructose.
And all of that
adipose
uses
fructose.
ATP.
Yeah, it depletes it so quickly, and it leads to degradation of AMP into uric acid as a result. This is like a metabolic pathway. Your body essentially thinks it's in starvation mode even though it's feeding, so it leads to a little bit of insulin resistance because, for example, you want to preserve glucose for the brain, but as you mentioned, insulin resistance isn't great, especially in other areas of the body. You mentioned like in the placenta, in my case adipose tissue. You get vasopressin release, so it increases blood pressure.
Right.
I wonder how that would affect something like preeclampsia. And you're also stimulating more fat storage and glycogen storage, surprisingly. So a lot of different effects. It causes even a hypoxic response. So you're decreasing mitochondrial function while favoring glycolysis, so reducing oxidative respiration.
Right. You're going anaerobic.
The issue is beta oxidation is an oxidative process, and it occurs in the mitochondria. So, in a sense, fructose metabolism is not great for a condition where you're accumulating too much fatty acids in the liver. I think fructose insulin also happens even normally with something like starvation and stress. I don't know. Did you read into that at all? The role of fructose in other organs?
I looked at fructose in the placenta. And so basically it's that same idea of the body's in starvation mode, right? And you're switching over into the anaerobic energy production, like glycolysis, except your body can also create fructose, and the way that it does that is through aldose reductase. And aldose reductase's activities are stimulated by ischemia and hypoxia. So if you have those two things happening because your placenta is not getting enough blood and not getting enough oxygen, the aldose reductase is kind of activated and it's creating more fructose. And then fructose comes down to uric acid that can cause endothelial damage. And that endothelial damage is such a big hallmark of preeclampsia. So I think it maybe makes preeclamptic issues worse.
Yeah.
Right? When you have preeclampsia, you're trying to avoid as much endothelial damage as you can because it's already happening. So it's just one big cycle where, like, this contributes to endothelial damage, this contributes to endothelial damage. And it all comes down to, like, placenta not getting enough blood.
Yeah. Don't drink. I think you can see on the back of ingredient labels, high fructose corn syrup.
Yeah.
Everyone says it's bad for you. Even more reason.
It should have been our biggest takeaway from our station.
Yeah. Even increases fructose, increases the amount of intestinal permeability, so allowing things like LPS endotoxin to infiltrate into circulation. If I can just touch on that really quickly, especially in NASH, when you have inflammation too, that also increases intestinal permeability. There is a big link in terms of gut microbiome and steatosis. So I started a school study published in Gut in 2013 by Leroy and colleagues, where they sort of used weight-matched obese C57 mice, and found that if you did one of these transplants, like the fecal microbiome transplant, essentially take the stool of a mice that was that had a steatotic liver, like fatty liver, and gave it to one group of mice that was sterile, and then gave healthy mice, also obese, to another group of mice So one has steatotic stool that's been transplanted, another one has non-steatotic stool transplanted in them. They sort of mirrored those conditions. And one interesting way they thought this might have been, or this is from another study, I think, I'm sort of trying to connect the dots. There's 2 strains of bacteria here, S. cerevisiae and W. confusa, they actually produce ethanol and acetaldehyde under high fructose conditions.
Oh.
So they're producing like alcohol. So maybe
Wait,
this is
is
non-alcoholic
wild.
fatty liver, but alcohol from bacteria could be driving pathogenesis.
Yeah.
So I think part of it does start in the microbiome. That's just one aspect of it. But going back to LPS and endotoxin, LPS and endotoxin activate your immune cells a lot of times, like Kupffer cells, hepatic stellate cells, and those are the things that drive fibrosis and inflammation.
Right.
So I feel like there's a big connection there in terms of, especially if you're getting infiltration from your gut. Depending on what your gut is comprised of can make a big difference. Depending on what metabolites getting into your circulation can make a difference. And also, increasing permeability leads to a lot of those inflammatory mediators getting into circulation and activating things like your Kupffer cells.
Yeah, I think in terms of like preeclampsia, I haven't looked super deep into it, and I think it's also like a fairly new area of research, but they think that like the microbiota in the gut also could play a role, which like I would never even have thought. But basically the study that I read, they were doing, your study was mouse to mouse fecal transplant. Okay, mine was human to mouse fecal transplant.
Oh.
Yeah. And so
keep talking.
basically they went from preeclamptic patients, did a fecal transplant into normal pregnant rats and preeclamptic rats. And in the normal pregnant rats, it caused increased blood pressure, which is like a big thing in preeclampsia, but not any of those other major symptoms of preeclampsia, like inflammation or like improper spiral remodeling. But when they did the fecal transplant from preeclamptic humans into preeclamptic rats, it caused higher blood pressure and inflammation. And then they were like, okay, let's try it from normal pregnancies. So they did it from people with normal pregnancies into preeclamptic rats. And it actually had like a protective effect. Like it relieved symptoms of preeclampsia. And like even spiral artery dysfunction, it like helped with that, which is like wild. And I wonder, they didn't really give a great reason for why. They basically said that like various levels of propionic acid and like butyric acid are reduced in people with preeclampsia.
Are those like short fatty acids?
I think so.
Bacteria produce a lot of signaling fatty acids.
Yeah. So it's less short chain fatty acids.
That have protective effect.
Yeah.
Interesting. I think with like medium and longer chain fatty acids, that's when you start getting into like pathogenic increase. I think short chain fatty acids are generally protective.
Yeah. But that's interesting. Like I wonder if that could be a root of like
we're all gonna be getting
fixing preeclampsia.
You heard it here. We're all gonna be getting fecal transplants soon.
Fecal transplants are actually so cool. Like they actually work.
The RCT volunteer.
So basically they think that like gut dysbiosis could kind of aggravate your intestinal barrier damage and like contribute to preeclampsia development because you don't have the protective effect of the short chain fatty acids. But like, I don't know. I feel like it was a big claim to be made by like one paper. And they were trying to make a claim between having these decreased levels of propionic acid and butyric acid in fecal serum and placenta and they were like, it correlates. So, like, therefore, somehow it's getting from your fecal matter to your placenta. And, like, maybe, but, like, I don't know.
It's such a high-order study. Like, it's like correlation doesn't equal causation.
Yeah. Yeah, I will, you know, maybe down the road they'll do some transplants in humans and see.
Yeah.
Maybe we'll have more convincing evidence.
Mm-hmm. Mm-hmm. Yeah, a lot of interesting stuff. I think, what have we talked about? Fructose, microbiome.
Mm-hmm.
I think going back to diet, and we talked about fructose, and we talked about how that affects gut microbiome. That's a reciprocal relationship. We touched a little bit on ethanol production by microbiome. I think it's interesting. With the liver, and I'm wondering how ethanol sort of travels through the placenta, what the cause is there. The liver is especially prone to damage from alcohol. You have alcoholic fatty liver disease. So I can tell you a little bit about how that happens, and then I'd love to hear more about whether there's any connections there.
Yeah.
But really in the liver, alcohol mainly increases oxidative stress, activates cell death pathways, and downregulates normal lipid metabolic pathways. I think those are probably the three ways it causes damage. So normally alcohol is broken down into acetaldehyde by alcohol dehydrogenase. And that acetaldehydes usually should be converted to acetate, but it's that middle intermediate, the acetaldehyde, that's causing a lot of the damage. It's coupled with NADH production, but every time it does that, you're inhibiting another enzyme I was reading called xanthine dehydrogenase, and the problem is when you inhibit xanthine dehydrogenase, you're actually oxidating xanthine, and that produces reactive oxygen species. This was at least one way, which this coupling with NADH production was creating reactive oxygen species. As a whole, when you create a lot of NADH, you're at the same time driving fatty acid production, lipid production. So you're tending to store more fat. So that NADH production causes its own damage. Also, ethanol and acetaldehyde block DNA methyltransferases and acetaldehyde itself, creating these reactive oxygen species, like I was mentioning, can bind with DNA as an adduct. So you're getting these sort of modifications and blockage of normal DNA processes.
Right.
When you start messing with DNA, your cells start realizing, oh, okay, we need to kill this cell, essentially. Like if you get a breakage, but you block all these proteins now, all of a sudden it's no longer a viable cell. Ethanol also, or at least acetaldehyde, causes CD95 or fast ligand expression. So that's on hepatocytes. That's signaling for death signaling, essentially, and mitochondrial membrane permeability. These are probably also byproducts of just reactive oxygen species production. Mm-hmm. But so you're essentially creating inflammation and activating cell death pathways. That's one way. The other way is ethanol blocks AMP kinase. So AMP kinase is a really key transcription factor, hugely involved in lipid metabolism. When you inhibit it, you reduce PPAR alpha activity. PPAR alpha is another transcription factor which expresses a lot of the proteins related to beta oxidation. You're also upregulating SREBP1, which is sterile regulatory element binding protein. Essentially, it's this enzyme that produces triglycerides in the liver. So then the plasma reticulum helps produce lipids, and you can't produce as much VLDL, which I said earlier in NAFLD. Right. It's an issue because you can't get the lipids up. So it's screwing up your cell's ability to not only break down fat, but also to actually survive. So it stimulates more of that necrosis and death signaling. Not good.
Right. So is that where you end up with cirrhosis?
Yeah, so alcohol is it used to be one of the main causes. I'm not sure if NAFLD has overtaken alcoholic fatty liver, but that's why you end up getting into cirrhosis. Or it's really a byproduct of the lipotoxicity.
Oh, okay,
Because
I see.
lipids build up.
Right.
But this is probably worse than NAFLD because it's not just the lipids causing damage, it's also the alcohol itself directly signaling for inflammation and ROS reduction. So, I'm not sure. How does ethanol play a role? Does it pass through the placenta?
It does, yeah. So it can go, it like just straight up diffuses across. But basically your like fetus liver, it's not developed enough, and so it just accumulates in the bloodstream. So that's bad. Basically, it can like reduce offspring size and cause, what's it called when...
Oh, fetal growth restriction?
No.
Is that?
Yes, but fetal alcohol...
Oh, fetal alcohol syndrome?
Yes. Is there a D attached to it too? Is it FASD? Fetal... Is it just FAS?
Fetal alcohol syndrome. I know it's FAS.
Okay.
FASD.
FASD?
Spectrum disorder.
Oh, okay. So yeah, basically, fetal alcohol spectrum disorder. But I was really interested in like the effects of alcohol more on placental function. And so basically, it's like exposure to alcohol during pregnancy, it's associated with reduced placental weight. And so that reduced placental weight is probably because of alterations in placental vasculature and like hypoperfusion of the placenta. So the placenta is not getting enough blood to grow to the size that it needs to actually be. Like probably less density of the blood vessels. And they know this because of smaller placentas, but also when they do immunostaining, there's lesser levels of placental growth factor and vascular endothelial growth factor, and you need both of those to form the blood vessels in the placenta in the first place. So that's pretty interesting. And then also, you know, insulin-like growth factor, which is really important for like paracrine autocrine signaling to like aid the tissue growth and the development of the placenta. And it also has anti-inflammatory functions, so it's like super important in terms of like placental development. Chronic exposure to ethanol will actually decrease the levels of that factor.
IGF-1. Right?
Yeah. And it also increases the amount of inhibitory IGF binding proteins. So IGF is usually found in the blood bound to a different protein. And so with like chronic ethanol exposure, the protein that it tends to bind to actually inhibits its function. So even the IGF that, like you have decreased IGF to begin with, and the IGF that's left in the blood is being further inhibited.
I wonder if there's just like less of that protein because the ethanol might be damaging the liver. Like there's a good chance that binding protein is produced in the liver.
Yeah, probably. And then also ethanol exposure can inhibit insulin, and so that loops back to the angiogenic functions of insulin in terms of placental growth. So that means that like drinking alcohol, it doesn't just affect the fetus, but it also affects like the placenta and the development of the placenta, and then that affects pregnancy experience down the line. And basically you need IGF-1 to stimulate glucose uptake in the trophoblasts, and it also regulates placental hormone secretion. So if you have less IGF-1 because of ethanol exposure, then you're getting issues with glucose uptake, you're getting issues with placental hormone secretion, which was bad, obviously, because you need all of those hormones in order to regulate placental function. And then even when you're breaking down the ethanol, it's like what you said with the reactive oxygen species, right? Because it's broken down by ethanol hydrogenase. Yes.
Alcohol dehydrogenase.
Yeah. Alcohol dehydrogenase. And then also by CYP2E1. And I think it's especially CYP2E1, when it metabolizes ethanol, it creates those reactive oxygen species. And then they talk about preeclampsia as being a state of oxidative stress, and that maybe one of the contributing factors is an inability to deal with oxidative stress. So if you already have like an inability to properly address reactive oxygen species, then that could increase preeclampsia effects, or increase risk for preeclampsia, which is like, wow. Like everything, I feel like, increases risk for preeclampsia. Like everywhere I look.
This is why I think like the second insult doesn't have to be so specific. There's not just one cause.
Yeah.
Like it's really just something that
It
probably
could be
causes
like
inflammation.
literally anything at this point.
Or like activated. Have you heard of? Did you come across that term, like activated trophoblast? Because what cell type release is the syncytial trophoblast? The cell type that's releasing the cytokines.
Yeah.
Okay. Have you come across like activated syncytial trophoblast in your research? So I wonder if there's just like this activated phenotype in syncytial trophoblast, and if there's different causes that can cause it. Like they can, I guess, like, maybe poor remodeling leads to ischemia, leads to a phenotype, and then it's like a phenotype that's prone to just like waiting for a second signal.
Yeah, they do talk about like just like some people might just be more susceptible to.
Maybe it upregulates like TLRs or something.
Yeah, like I don't know actually.
I don't know.
That's kind of, I don't know. It's like everything.
Maybe.
I feel like it's all these factors just coalesce.
Yeah. I wanted to talk about more of the direct connection, I guess, between the placenta and the liver, which is the HELLP Syndrome.
Oh.
I thought that was super cool. That's one of the first things I came across.
Yeah, right at the beginning I remember you were like, HELLP Syndrome, and I was like, I gotta back up, like.
It's very cool. It's more indirectly affecting the liver, from what I know. At least this is what I thought, and then I see that no, it actually does directly affect the liver as well. Do you want to give an intro?
Yeah.
To HELLP?
So basically, HELLP Syndrome stands for hemolysis, elevated liver enzymes, and low platelet syndrome. And I think it's generally, in a lot of literature, it's generally seen as this like very severe form of preeclampsia, or like a severe complication of preeclampsia. But I did read that sometimes you can get symptoms of HELLP without like proteinuria and hypertension. And I saw that and I like tried to click on for like reference to see where it would lead me, and I like literally couldn't find it. So like I believe it because that's what I heard in class too, but I don't know why. I don't know what it is that can cause HELLP syndrome without preeclampsia. Basically HELLP is severe preeclampsia, right? Hemolysis.
That's what people think.
That's what people think. And that is, I'm still investigating that because I've seen it in a couple articles and I can't find kind of an explanation for why it occurs outside of preeclampsia sometimes.
They're just correlated.
But a lot of times preeclampsia develops into HELLP. And HELLP is where you have hemolysis, so like your red blood cells are basically getting shredded. And then elevated liver enzymes and a low platelet count. And like basically you're having endothelial injury, and maybe that's where preeclampsia tends to develop into HELLP is because it has that endothelial injury aspect. And
like
that...
from inflammation?
Yeah, from the inflammation. And that causes...
From hypertension to more shear, which is force on the endothelium as well.
And so that, like, it exposes the collagen that's under the endothelium. And that kind of triggers, like, a coagulation response. And so you form all these blood clots, which is using up the platelets in the blood. And that's where you get the low platelets. But basically, you're creating all of these little thromboses, basically, like tiny little clots in the blood. And as your blood passes through the vessels that have been damaged and have formed these clots, they get shredded. And so that's where you get the hemolysis and the lower platelet count. And then in terms of the liver enzymes, it's just basically all of these come back to the liver. And it causes liver damage. And as the liver dies, it, like, starts releasing the liver enzymes.
Yeah. Just going back to the coagulation, I think it's really interesting to talk about von Willebrand factor. One of those proteins that's upregulated when you have activated endothelial cells.
Yeah.
It's what catches. It's like this, like, mesh-like protein catching platelets to cause clotting.
Right.
That's what's shearing those blood vessels that are going through it. It's like fibrin networks.
Right.
That your red blood cells, your erythrocytes aren't meant to go through. And you have less, like, proteases like ADAM-T13 to cleave some of that fibrin. So it's just built up and leads to hemolysis, like you were saying.
Yeah, and you're getting, like thrombotic microangiopathy, where this damage is happening in those tiny little capillaries. And so your blood vessels, or your blood that's going through the vessel, is already traveling kind of like one by one. And then you're making that hole even smaller.
Exactly.
Because you have these blood clots and you have the fibrin. And so it creates a really good environment to be shredding blood.
And it depletes your platelets, so you get the low platelets. I think also, there's definitely ischemia in the liver, which can lead to, I guess, hepatocyte death, necrosis, and elevated liver enzymes. But there's also just placenta-derived fast ligand a lot of the times, especially in the context of preeclampsia. That's you might be getting some of that CD95 release from the placenta.
Right, and that it triggers TNF alpha, right?
It causes death signaling in hepatocytes.
Yeah.
But I think there's also TNF alpha released from other inflammatory factors released from the placenta. I was reading even SFLT1, that soluble VEGF receptor.
Right.
I would assume it's more indirect. I think the paper I was reading, it wasn't too specific, but it's likely just reducing VEGF signaling in that area. Probably related indirectly to eNOS production, or NO production from eNOS.
Right.
So overall, indirectly, it leads back to increased inflammation, probably leading to that hepatocyte death phenotype.
Yeah, and I read that the hepatocytes, they die without the fatty cell transformation.
Yeah.
They just, like, straight up necrotize. Necrose?
Undergo necrosis.
There's no verb for necrosis?
I don't think so. That's really interesting. But at the same time, you sometimes get HELLP, preeclampsia, and acute fatty liver disease.
Right.
So in pregnancy, there's this acute fatty, but that's usually due to a genetic condition, like a genetic mutation. Right. I think it's LCHAD.
Yeah.
They sometimes call it a trio. I guess like maybe LCHAD mutation is what causes some of that trio. But it's basically a mitochondrial protein that assists with fatty oxidation, fatty acid beta oxidation. So your fetus essentially can't break down, or the placenta, the fetal placental unit, can't break down fatty acids. And the maternal side is just shunting all these nutrients to the fetus. It's not being really broken down at all, even though the fatty acids are building up. So a lot of it ends up getting into circulation.
Right.
Depositing in the liver, which, as we talked about, isn't great at storing lipids. Probably inducing lipotoxicity in the long run. But I think it's a lot of the times fatal, right? Because
Acute fatty liver in pregnancy.
Likely from just also fatty acids building up in the fetal mitochondria.
Yeah.
So I thought it was an interesting connection between how sometimes you do get fatty liver in pregnancy. I wonder if in that case with LCHAD mutation and acute fatty liver pregnancy, whether it's the liver causing preeclampsia and HELLP through just liver failure, or like, I guess, slowly losing function of the liver. I guess not slow, it's like rapid accumulation of loss of liver function. Or if fatty acid metabolism does play a role in the placenta as well.
I wonder, because...
Remember, it causes all that inflammation in the liver, where fatty lipids build up. It's from undergoing too much beta oxidation in that case. But I guess maybe ER stress, if it has this fatty acid, it can't break it down. So it's trying to create triglycerides in the placenta, but too much of it leads to ER stress, and that's causing its own inflammatory proteins being released.
This is your personal theory?
This is not in the literature as far as I know. But I don't think there's any literature on this. Is such an understudied area.
Yeah.
I think this is where you can have expertise in two different topics and try to find these connections. This is why
they need interdisciplinary research.
Yes. I think it does make sense though. It could be that second hit.
Yeah.
Like there's aberrant placentation, but the second hit in this case may be from, impaired beta oxidation.
Yeah, I wonder if they talk about you need the second hit, right? And what if that second hit is just inflammation?
Yeah. And
it doesn't matter if the inflammatory factors are coming from the placenta because of placental, hypoxia. Like, what if you're having liver issues and the liver is releasing inflammatory factors? I know it's less systemic, like it's more localized, but I wonder if that would be enough to be the second hit to push someone over into preeclampsia.
Possibly, or indirectly even from good microbiome issues too, right? Like LPS we talked about.
Yeah.
Or even more indirectly from liver failure, toxins building up. Okay, I think we can wrap it up there. I wanted to just summarize maybe some of the things we talked about. We talked about a lot.
Yeah.
And I think it's interesting to see some of the connections, these almost like canonical connections between so many diseases too, right?
Yeah.
And then more specific ones. So 2 organs intertwined in filtration. It seems like metabolic syndrome plays a big role in both of them, though different roles.
Yeah, I think my biggest takeaway from this is less that the placenta and the liver are so intertwined, but there are so many different factors that affect a lot of different areas in the body. Because you look at something like metabolic syndrome, and yes, they both affect liver and placenta, but they probably affect a lot of different organs as well.
That's true.
And so I think more so than the liver and the placenta are connected, we found a lot of different factors that have effects in a lot of different parts of the body.
Just common, like I say, like canonical areas where things go wrong a lot of times. Like the gut microbiome connected to so many organs. Nitric oxide, vascular dynamics, the balance I think between eNOS and iNOS in my case, but just needing some nitric oxide production from eNOS.
Right.
I think you said AKT, activation.
Yeah. Like that relates to insulin resistance. Yeah, I think it's wild how interconnected the body is, and that it's hard to look at it as this organ, this third organ, because it's one system at the end of the day.
Mm-hmm.
And so everything that you do to your body, or like things that go wrong in your body, it doesn't affect just one thing. It ends up affecting the whole system.
Yeah. They all end together, and apparently in inflammation.
Yeah. Everything ends in inflammation.
Yeah.
Yes. Do we have oh, actually, I was going to ask you if you could continue taking this course, because we are at the end of the course.
Mm-hmm.
Say the course went on into like another semester.
Yeah.
What would you want to look into? What do you feel like you didn't get the chance to cover this time?
Oh.
Your further sources of inquiry.
That's a good question. I think something I found really cool, I personally didn't study as much because I started looking into it for my other class, Pathophysiology, is congenital heart defects and the surgeries people do to sort of treat them. Oh my gosh, that's one of the coolest things I learned. We had a lecture by Dr. Dragan Perdescu, calling in from Romania. He was telling us all these different ways that cardiac surgeons are like butchers to the heart. Like
Wild.
They save a lot of lives. Yeah. But congenital heart defects for sure, and especially how you have all these unique surgeries to sort of help fix what's wrong with the heart. Mm-hmm. Definitely very anatomical in nature, which I like. I think a lot of our research was a good mix of both, but definitely more on the physiology side, things that are hard to see. Mm-hmm. What about you? That's a really good question.
I think I would agree, like, in terms of looking at fixing problems. Because I think this time around, I really focused on things that can go wrong with the placenta.
Yeah.
And, like, the issues of preeclampsia. And so I would be interested in seeing how different cures work or really have failed. You know, I think it would be interesting to kind of explore how do we, like, fix these issues.
Right. But still in the realm of, like, preeclampsia.
I think so, yeah.
Yeah.
Because now I'm so invested in the problem, I want to know how it can be cured.
It's a cool black box. NAFL's less interesting.
How can it be cured beyond delivering? Yeah.
Good solution. Okay.
Right.
That was a good discussion. Thank you for having the conversation with me.
Oh, thank you for having this conversation with me.
And thank you to the 3FO3 teaching team and the peer tutors.
Yeah. Thank you for listening to this behemoth of a podcast. We're at 1 hour and 46 minutes right now, but we'll try to cut some of it down.
Thank you so much for listening, and we hope you enjoyed. See ya.
Bye.